Bacterial nutrition and growth. We'll be considering bacterial nutrition and growth, nutritional requirements, and environmental factors, phases of bacterial growth on a growth scale. Bacterial nutrition. Bacteria are devoid of chlorophyll and they perform heterotrophic mode of nutrition. However, there is a small group of bacteria which are autotrophic in nature. That means they manufacture their own food. They don't depend on other organisms for survival but manufacture their own food through photosynthesis and also chemosynthesis. Autotrophic bacteria include green sulphur bacteria. They are strictly anaerobic photoautotrophs. Their photosynthetic pigment, chorobium chlorophyll, is located in the vagination of plasma membrane towards the cytoplasm. They uses hydrogen sulfate. Light splits this hydrogen sulfate and forms hydrogen which combine with carbon dioxide to form formaldehyde. Sulfur is extracted out and deposited extracellularly. Example of this green sulfur bacteria can be a chorobium peridiction, etc. So here, these bacteria, they can use this hydrogen sulfide or other reduced inorganic sulfur component as a donor of hydrogen. As seen in the equation there, see how bacterial chlorophyll, in the presence of light and bacterial chlorophyll, the CO2 combined with the hydrogen sulfide and water to produce the formaldehyde plus the tetraoxosulfate 6 acid. And also here down you can see like that how they occur in light and bacterial chlorophyll. Example of okay, so chemosynthetic autotroph like sulfur bacteria, they had this hydrogen sulfate here combined with the oxygen to give you the sulfur compound and also the water plus energy produced. But here light is not needed. So another type of which is utilized from sulfur. Also the ion bacteria you can see like that. So differences between photosynthesis and chemosynthesis is that the photosynthesis takes place in the presence of light, but chemosynthesis, as I already said, does not require light. So reaction of photosynthesis are endothermic, while that one of chemosynthesis reaction are exothermic. And you can see that the energy available in photosynthesis is more than that of chemosynthesis. The energy available in chemosynthesis is much less than the photosynthetic energy available. So the differences between the photosynthesis and chemosynthesis is clearly defined. So you can see here the presence of light indicates photosynthesis and the presence of chemicals also indicate chemosynthesis. So heterotrophic bacteria, these bacteria obtain their food from any organic source. There are two types, which is the saprophytic bacteria and parasitic bacteria. So, sorry for that. So, these bacteria in saprophytic grow on decaying organic matters and live by digestion and absorbing them. Growth of bacteria. The growth of bacteria requires proper nutrition and proper environment. So the requirements of nutrition in bacteria are so simple, you can understand them when you just pay attention. Let's just go ahead with them. So first is the essential elements. Essential elements like the nucleic acid, the carbohydrate. You know that essential elements require photosynthesis of structural compounds such as nucleic acid, proteins, fats, carbohydrates, or carbon. They are all the essential elements needed by bacteria, and also mineral sources. Mineral sources like potassium, iron, carbon, and also know that these are mineral sources essential. They're also essential for growth of bacteria, so they are also very, very important for bacterial growth and nutrition. And also the organic growth factors, these compounds are called vitamins. and they're required at micro level as essential. So here, these organic growth factors are required in micro levels. That is, they're not required too much at large in the body, but when they're split down into smaller particles, they are needed in the body. So you also have the requirements of proper environment. That is, proper environment where organism, these bacteria can grow without any effect on them. So these factors include water, oxygen, carbon dioxide, temperature, pH, light, and osmotic pressure. So water is an essential requirement for growth because approximately 80% of cell volume consists of water, and also oxygen. Bacteria differ in requirements of oxygen. There are two types, the aerobes and anaerobes. The aerobes require oxygen for growth, while the aerobes again can be divided into obligate, that is the pseudomonas, or the facultative anaerobes. So that is, they are normally aerobes but can grow without oxygen. Example of the vibrio curare, the anaerobes do not require oxygen for their growth and they may die in presence of oxygen at Clostridium. Carbon dioxide also is needed very in small amounts, and they require for all bacteria, which may be available endogenously or may be collected through atmosphere. Temperature also needed at a level of 25 to 40 degrees Celsius for thermophiles like that. So thermophiles are also organisms that survive in deep water sometimes. The pH can also affect. So the pH here also, bacteria associated with human disease require pH of 7.2 to 7.4, but vibrio curare, for example, require a pH of 10.5, like that. So bacteria like lactobacillus and therobacillus thalassanes require the pH of 3.0 for their growth. And also the light, bacteria generally prefer to grow in dark, but some phototrophic species grow in light. So like the osmotic pressure, one of the factor, bacteria are able to grow in a wide range of osmotic pressure because of right and top cell wall. Bacteria are autotroph and heterotrophs in nature and can grow when environment it found is favorable for them. So as you can see here, the depth in meters and also the number of colonies, the type of species of bacteria found in that particular environment. Like the species here is the Pranococcus cereus, you know that the number of colonies is 49, depends on the environmental conditions of that particular area. You see this, the 100, see 32, 24, 35, and 30. Here you see that it depends on the particular place where the organism, precisely the bacteria we're talking about, is found. So if the absence of oxygen, the absence of water and all the environmental factors, if the organism is lacking, they cannot produce effectively. So the presence of water, presence of oxygen, presence of normal temperature, and also the presence of low but not all that pH, but if the pH is very high, it cannot grow effectively. So the environmental factors are very essential and important in the growth of bacteria. So bacteria here can grow when and only when the environmental factors are effective for them. If the environment is not favorable for them, they can't grow. That means that the graph or the growth curve of this bacteria can lead to a death phase. Looking at the graph there, you see that at log phase the organism started growing because they entered the environment where there is the unoccupied environment, so they started growing. So you see that at log exponential phase there, you know that because of the presence of all the environmental factors, they grow exponentially. At the stationary phase, you see that this growth enters the carrying capacity of the environment. So the carrying capacity is the number of bacteria the environment can support. So when it enters the stationary phase, that means that the number of death and the birth is the same. So that is the equilibrium state. But looking at the graph critically, you see that it started decline. That is, the bacteria in that environment, because of the unfavorable conditions of the environmental factors, is the bacteria started dying off. So when they are dying off, that means that the population of that bacteria is reducing as a result of unfavorable condition of the environment. So the lack of water, the lack of oxygen, all the environmental factors affect the growth of bacteria. Thank you guys for watching my video.